Hello, this is Seher from Easy Peasy, and the topic we are going to discuss today is called as transcription of messenger RNA in eukaryotes. Eukaryotic organisms are those organisms in which we have true or well-defined nucleus present inside their cell. The transcription is a process in which DNA produces RNA from it, and that process is called as transcription. Unlike prokaryotic cell, in which the process of transcription and process of translation is occurring simultaneously, in eukaryotes the process of transcription occurs inside the nucleus. Then this messenger RNA will come out into the cytoplasm, and here the process of translation will start. Inside the eukaryotic cell, the DNA is present in highly compact structure called as chromosome with the help of histone proteins. So, DNA exists in two different forms, that is euchromatin state and heterochromatin state. In euchromatin state, the DNA is loosely coiled upon itself on the histone proteins and is available for the transcription machinery to attach itself to it and start the process of transcription. On the other hand, heterochromatin state is a highly compact structure in which the DNA is tightly packed around the histone proteins and is not available for the process of transcription. So, if we want a gene that needs to get transcribed and it is present in heterochromatin state, it needs to shift itself into the euchromatin state in order to transcribe itself. So acetylation is the main modification that turns the heterochromatin state into the euchromatin state. As we know that DNA has a negative charge and histone proteins have positive charge, that's how they are coiled upon itself. So acetylation basically neutralizes the positive charge present on these histone tails. So these acetyl groups attach itself to the tails of histone proteins, neutralize the positive charge, and then DNA can lose itself around the histone proteins and shift itself from heterochromatin state into the euchromatin state. The enzyme that is helping the attachment of acetyl group with the histone tail is called as histone acetyl transferases. And to shift it back into the heterochromatin state, we have another enzyme, and that is called as histone deacetylases. We have certain other proteins that also helps in the shifting of heterochromatin state into the euchromatin state, and these proteins are SWI/SNF, NURF, and SWR1. We are not going to discuss the details that how these proteins are shifting the heterochromatin state into the euchromatin state, but we will make a separate video on that. We have other type of modifications as well that is going to activate the process of transcription. Here you can see that we have an activation mark and we have 4 histone proteins, and that are H2A, H2B, H3, and H4. To activate the process of transcription, H2B needs to get modified with ubiquitination. Acetylation should be present on H3. Di or trimethylation should be present on lysine, that is positioned on number 4 on H3. Acetylation should be present on H4. If that is the code present on histone tails, then these modifications turn on the process of transcription. On the other hand, we have inactivation marks as well. That is, if ubiquitination is occurring on H2A instead of H2B, the methylation is present on lysine number 9 instead of lysine number 4, and 3-methylation is present on lysine 27 on H3. If that is the code present on histone proteins, then it is going to order the transcription machinery to halt the process of transcription. Now let's discuss the structure of eukaryotic DNA. So here we have control site. In that control site we have regulatory promoter and core promoter. Regulatory promoter have 2 consensus sequence here, that is GC box and CAT box, and core promoter have TATA box. Keep in mind that all these consensus sequence is not necessary to present in every eukaryotic DNA. Sometimes we have GC box in the sequence but not the CAT box. Sometimes we have the CAT box but not the GC box. Sometimes we even don't have the TATA box, but we have a core promoter site on which a protein will attach itself. Then we have structural gene site in which we have exons and introns. Exons are that part of DNA that's going to code for a protein. And introns are that part of DNA that is not going to code for any protein, so they are the non-coding areas present inside the DNA. Now they are going to make a messenger RNA, and this messenger RNA will only make one protein from it. If a messenger RNA can only code for one protein, that type of messenger RNA is called as monocistronic messenger RNA, and that is usually present inside the eukaryotic cell. Now, only one protein cannot start a single biochemical pathway, so we are not having any operon in this situation. In eukaryotic cell, we have different types of RNA polymerases as well, because each RNA polymerase is going to transcribe a different type of RNA. Here in this picture, we have three different types of RNA polymerases, and that are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3, and they are represented by this big circle in each picture. Now these RNA polymerases are attached with tiny different other proteins, and these proteins are called as transcription factors. Each RNA polymerase have their own transcription factors. These transcription factors help RNA polymerase to get attached itself to the core promoter site. Now each RNA polymerase have their own promoter site as well. Just like in the first picture, the RNA polymerase 1 is going to attach itself to this UCE and core promoter site that is called as UBF dimer. For RNA polymerase 2, the recognition site is TATA box. For RNA polymerase 3, the recognition site is A or B sequence. Now RNA polymerase 1 transcribe 28S, 18S, and 5.8S ribosomal RNA. RNA polymerase 2 transcribe messenger RNA, small nuclear RNA, small nucleolar RNA, and microRNA. RNA polymerase 3 is going to transcribe transfer RNA, 5S ribosomal RNA, some small nuclear RNA, and small nucleolar RNA. We are just focused on messenger RNA, and that is going to transcribe by RNA polymerase 2. So we will only focus on RNA polymerase 2 in this video. Now, the DNA strand that is going to act as a template for RNA polymerase is called as non-coding strand or antisense strand. The RNA that is coming out of this DNA is called as primary transcript, and it is always from 5' end to 3' end. In eukaryotic transcription, we have 5 different stages, and that are activation or assembly stage, initiation stage, elongation stage, modification stage, and termination stage. The DNA sequence that come under the category of activation and initiation is called as upstream sequence, and the sequence that comes under the category of termination is called as downstream sequence. Now let's discuss each stage in detail. So the first stage is called as activation stage or assembly stage. Now I'm taking the same genome that we discussed before, and this part of the DNA is called as +1 site. Now this +1 site is basically the site where the transcription will start. If I go towards the right side of this +1, we will have +2, +3, +4. And if I go towards left side of this +1, I will have -1, -2, -3, and so on and so forth. So TATA box is present on -25, CAT box is present on -80, and GC box is present on -100. We also have another sequence called as enhancer sequence that is present 100 or 1000 base pairs upstream from the transcription start site. Now, The TATA box is going to come under the category of core promoter site, and GC box and CAT box plus the enhancers is going to come under the category of regulatory promoter site. So regulatory promoter is a kind of contract that needs to get signed before the core promoters start the process of transcription. Now, to attach RNA polymerase II to the core promoter site, we need 4 different types of proteins here, and that are transcription activators, architectural regulators, co-activators, and basal transcription factors. Initially, the transcription activator is going to bind itself to the enhancer region. We have 2 specific type of activators as well that is going to bind itself to the regulatory promoter consensus sequence. One is called as SP1 that is going to bind itself to the GC box, and the second one is called as CTF1 that is going to bind itself to the CAT box. The function of these activators is to provide signal to the core promoter or transcription machinery to start the process of initiation. The second type of protein that we are going to talk about here is called as architectural regulators. Now these regulators are going to bind itself to the DNA and is going to bend the DNA like this, and it helps the enhancer region to come in the close proximity with the core promoter region so that they can easily give the signal to the core promoter to start the process of initiation. These proteins are called as high mobility group or HMG proteins. The third type of protein that we are going to talk about is called as co-activators. These co-activators are also called as mediators. Now these co-activators will not make a direct contact with the DNA, rather they will make a contact with the activator and become a kind of a bridge between the transcription machinery and the enhancer region to provide signal to the core promoter so that they can start the process of initiation. The basal transcription factors basically involved in the initiation stage. So let's discuss the initiation stage now. Initiation. Okay, in the initiation we have 2 main proteins that are involved in this process. One is the basal transcription factors and the other is the RNA polymerase II. The basal transcription factors that are involved in the process of initiation are TF2A, TF2B, TF2D, TF2E, TF2F, and TF2H. RNA polymerase have a tail type structure that is called as CTD or carboxy terminal domain. Initially, TF2D is going to go and bind itself to the TATA box. Now here's the thing. This TF2D has a TBP here. TBP is basically a protein that is called as TATA box binding protein. So if we have a TATA box present inside the DNA, TBP is going to recognize it and bind itself to it, followed by TF2D. But if we have a DNA that doesn't have TATA box, then TBP is not going to come to bind itself to the TATA box because it is not available. Only TF2D is going to come and attach itself to the core promoter site. The second protein that is going to help the binding of TF2D with the core promoter site is called as TF2A. So TF2A basically provides stability to TF2D and TATA box binding. The third protein is TF2B. Now TF2B is here and it is calling TF2F to attach itself to RNA polymerase II and bring it to the site where it can attach itself to the core promoter. Then to provide the stability to this transcription machinery, TF2E is going to come there, and then in the last we have TF2H that also have the helicase activity to open up the double strand DNA and start the process of initiation. Now let's start the process of elongation. In elongation stage, the TF2H is going to perform its function. Now TF2H have two different functions here. The first function is that it has a helicase activity, which is going to open up the DNA double helix like this. The second function of TF2H is that it has a kinase activity, which is going to phosphorylate the serine amino acid present on position 5 on the CTD domain of RNA polymerase II. When RNA polymerase II is phosphorylated like this, it can leave the core promoter site and move along the DNA template strand. When RNA polymerase II move along the DNA template strand, most of the basal transcription factors remove itself from this RNA polymerase II, and only the TF2F is present on RNA polymerase II in elongation stage. Now, the process of elongation in eukaryotic cell is not smooth. The RNA polymerase II can take several different pauses while moving along the DNA template strand, and these pauses can be from seconds to several different minutes. These pauses are called as checkpoints or promoter proximal pausing areas. Now, at this position, at this checkpoint, we have a teeny tiny messenger RNA here that is coming from 5' end to 3' end. To move this RNA polymerase II from the checkpoint, we need the help of several different proteins named as transcription elongation factors. The first elongation factor is ELL. It is going to come and attach itself with RNA polymerase II and suppress the transient pausing state of RNA polymerase. The second type of protein that is going to help is called as DSIF. Now this DSIF locks the RNA polymerase clamp into the closed state to prevent the elongation complex from dissociating. Now here's the thing. If this DSIF will come in contact with NELF, it will no longer promote the process of elongation. Rather, it will prevent the RNA polymerase to move further. Now, we need to move the RNA polymerase II. For that, we need to take help of another protein, and that is called as PTEFB. This PTEFB will come and attach itself with RNA polymerase II and phosphorylate the DSIF. When DSIF is phosphorylated, it can no longer make contact with NELF and it will remove from the site. Now, DSIF will help in the process of elongation. PTEFB will also phosphorylate the serine amino acid present on the CTD domain on position 2. Next protein that's going to come and help in the process of elongation is called as TF2S. Now this TF2S will come and bind itself with RNA polymerase and allow it to recover from the arrest position. The similar function is available for the last protein, and that is called as elongane SII, and this protein will attach itself to the RNA polymerase II and move it from the checkpoint. By this way, the RNA polymerase II will complete the process of elongation by passing several checkpoints. Modification. We have three different types of modification. The first type of modification is the attachment of 7-methylguanosine triphosphate on the 5' end. That is also called as the capping of messenger RNA. The second type of modification is the attachment of poly A tail at the 3' end of messenger RNA. The third type of modification is the spliceosome. It means that we need to remove the non-coding part of messenger RNA called as introns and attach the exons together and make a mature messenger RNA. During the process of transcription, attachment of 7-methylguanosine triphosphate is the first modification, followed by the spliceosome, and at the end we have to attach the poly A tail before the termination process will start. So let's discuss the 5' capping of this messenger RNA. 5' capping will occur at the very early stage of elongation when we have the primary transcript of about 20 to 30 nucleotides long. At this point, the cap synthesizing enzyme will attach itself with the tail of the CTD domain of RNA polymerase. Now this cap synthesizing enzyme will attach the 7-methylguanosine triphosphate to the 5' end. When 7-methylguanosine triphosphate will attach, the cap synthesizing enzyme will leave the CTD domain, and cap binding complex will come and attach itself with the 7-methylguanosine triphosphate during the process of elongation. The second type of modification is the spliceosome. So splicing of intron will occur during the stage of elongation. Here you can see that we are almost going to enter the termination stage, and we have a fairly big messenger RNA still attached to cap binding complex with the CTD tail here. Now, for example, these two portions are the introns present inside the messenger RNA. So this is intron 1 and this is intron 2. Now we have two different types of splicing here. The first type of splicing is self-splicing, in which the intron will make a hairpin type of structure and remove itself from it, like this. The second type of intron will need a helper protein that is called as non-coding small nuclear RNAs. Now these proteins are called as U1, U2, U5, U6. We also have U12, U11, but I didn't mention it here because I just need to tell you that this is the way the splicing will occur. Now these small nuclear RNA will attach themselves to the intron and will remove it from the messenger RNA like this. The third type of modification is the attachment of poly-A tail that will come under the stage of termination. So let's discuss the process of termination and the attachment of poly-A tail. Now here the messenger RNA we have has already modified with the spliceosome, and the attachment of 5' cap is already done here. At the stage of termination, on the DNA strand, we have a sequence that will signal the transcription machinery to start the process of termination. That sequence is mostly TTA-TTT. When transcription machinery reaches that sequence, it will code it on messenger RNA, and the messenger RNA will have AAU-AAA on it. Now this sequence will be recognized by a protein named as CPSF. The full form of CPSF is cleavage and polyadenylation specificity factor. Now this protein will attach itself to AAU-AAA. RNA polymerase 2 is not stopped on this termination sequence because all the elongation factors are still attached to it and it is still moving. So when it will move, it will be followed by another sequence, and that is called as U/GU rich region. Now this region will be recognized by another protein named as CSTF. The full form of this protein is cleavage stimulation factor. This CSTF will attach itself to the GU-rich region and will cut the region between AUAAA and GU-rich region like this. Now to attach the poly-A tail on the 3' end of messenger RNA, the polyadenylate polymerase will come and attach the poly-A tail residues. It will almost attach 80 to 250 A residues at the 3' end of this messenger RNA. The RNA polymerase 2 is still attached on the DNA template. So for that, we need to remove all the elongation factors that are attached to this RNA polymerase 2. For that, we need to remove this phosphorus atom that is attached to this DSIF. and this phosphorus atom was attached to it because of this PTEFB in the elongation stage. So to remove this protein, we need another protein called as 7SK small nuclear RNP. Now this protein will come and remove this PTEFB from the RNA polymerase II. When PTEFB will remove from the RNA polymerase, the phosphorus atom will also remove from the RNA polymerase. Now DSIF is without phosphorus atom, so it can attach itself back to NELF and halt the process of elongation. Now the RNA polymerase is not moving on DNA and is not making this messenger RNA. To remove this transcription machinery from DNA, we need 5' to 3' exonuclease here. When it's going to come and remove this leftover messenger RNA, all the transcription machinery plus RNA polymerase will dissociate from this DNA. And that's how the process of transcription will end here. Thank you very much for watching this video. If you like it, please subscribe my channel.